Thank you. Well, Ingemar and Ihai, thank you so much, and thank all of you for enabling me to come here, and I'm excited about joining your faculty tomorrow. Very much. It's a really tremendous honor for me to receive a degree from Karolinska, and given all that's happened here and what a wonderful history it's had. And I'm sort of an anomaly as a chemical engineer to be doing this, and I feel very privileged about that as well. So I've had the privilege also of giving lectures here before, so I wanted to do a little bit of a different lecture today. And the lecture really, it definitely does involve some science, but I'm not going to go into the science sometimes into the depth that I might normally. But what I was going to try to talk about is give you 6 examples and then some conclusions of how we've tried to create science in the laboratory. But one of the things that's been personally important to me, and I'll go over that, is I guess my frustration. You know, when I was starting my career about how, I published papers, and maybe it's the engineering person in me, but my frustration that I published papers, and I thought, very naively, that if I published papers, then, that would just lead other people to creating products. And that certainly did not happen. And I realized that, as I'll go over, that to possibly make that happen, I'd have to get involved in entrepreneurship myself. I should say as an aside that it's also been striking to me, and it's certainly not all my doing by any means, it's actually the doing of many people, but I was a graduate student at MIT in the 1970s. And in the 1970s, if you looked at MIT, the area around MIT, it was what we called the slum. It was terrible. There was no hotels, there were no restaurants, hardly any companies. There were some bars, that they call them bars here, so they were not very good ones, so you wouldn't want to go out at, maybe after 5 or 6 at night. So it was kind of dangerous. And what's happened, because of all these companies, is that in the last 30 years now, is the area around MIT has the highest concentration of biotechnology companies in the world. It's probably got a market capitalization of about $1 trillion or more. And there are all kinds of, maybe 10 hotels. Actually, most of the best restaurants are now near MIT. And there aren't any of those bars, I don't think. So it's actually been a remarkable transformation. And most important to me when I look at these companies is they're really creating things that are changing people's lives in cancer, multiple sclerosis, many other diseases. So I'm going to give you 6 stories today of things that I've been involved in, and then I'll try to just make some general comments about them. So the first story actually does start with what Ihai mentioned. When I was a graduate student, the normal thing for chemical engineers to do was to go to oil companies. And I could have done that. I had 20 job offers from oil companies. I always say 4 from Exxon alone. But I wasn't excited about that. And so then I tried to get jobs in education and then in medicine, and nobody would hire me. So I got 0 jobs and things like that. But then Judah Folkman was nice enough to offer me a position. And the position that he offered me was to see if we could isolate the first angiogenesis inhibitor. So it's kind of like basic research. But one of the key challenges, maybe the key challenge in isolating that inhibitor, was creating a bioassay. And if you look back at the history of medicine, that's often the case with new factors. Could you create a bioassay? And there wasn't one. And one of the challenges that we faced was that if we looked at any kind of organism, blood vessels don't grow that quickly. We wanted to at least be semi-quantitative. So what we wanted to have was a slow-release polymer that could deliver any of these angiogenic factors, which were fairly large molecules, like proteins and things like that. And we needed to be able to deliver it for at least a couple of months. And that didn't exist. People hadn't done that. And of course, there probably wasn't necessarily any reason to do that at that time other than something like this. But what would happen, as people may know, is that starting around that same time, the area of biotechnology and genetic engineering emerged. And when people tried to take large molecules by oral means, they really couldn't do it. They couldn't do it by a patch, and if you injected large molecules, you often had these very short lifetimes. So really, if you ever wanted to use them, for the most part, you'd have to have a way to deliver them in an unaltered form and yet protect them from harm. And that would also be true for the angiogenesis factors, at least in the assays we were using. So It turned out that Dr. Folkman was on the board of the one company that was involved in trying to do any type of drug delivery. I'll just show you a few things. So at that time, people in the literature said you couldn't use slow-release systems for, you could only use them for small molecules. And actually, when Dr. Folkman talked about this to the one company I mentioned that had done any work at all in a drug delivery system, ALZA, and they had some Nobel Prize winners like Paul Flory on their board in chemistry, but all of them said that large molecules couldn't go through polymers any more than we could go through a brick wall, in other words, walking. And I always say the only thing that I had going for me is I didn't know any of this. So I tried anyhow. I spent 2 years working in the laboratory, experimenting with different techniques, and I found over 200 different ways to get this to not work. But eventually, I was able to make little microspheres like this. Here's one cut in half. And we showed in Nature, we published the fact that you could use this kind of idea to release molecules of virtually any size for over 100 days. And later on, even using some engineering approaches, we showed that you could get constant release. But when I first got up and started trying to talk about this, I still remember there was a talk I had to give in Michigan 2 years after I started this, and I remember giving this talk, and I was 27 years old, and I rehearsed this talk for about 2 weeks because I was very nervous about speaking, and so I rehearsed the talk, and I got up and I gave the talk, and it was to a group of very famous chemical engineers and chemists. And when I got done with this 20-minute talk, I thought I did all right, but because I didn't forget too much of what I was going to say. And so I thought when I got done that all these older scientists and engineers, being nice people, would want to encourage me, this young guy. And when I got done, a whole bunch of them came up to me and they said, "We don't believe anything you just said." And in particular, they said that large molecules, like I said before, can't diffuse through these polymers. We were also using organic solvents, and they said, "Don't you realize that will destroy whatever you put in the system?" Now, actually, things actually went downhill for me after that. What happened is I wrote research grants to the government, and my first 9 were rejected very badly. And then I just wanted to and then I did decide that maybe I'd like to get a faculty position. But I applied to chemical engineering departments, and not a single chemical engineering department in the United States would hire me. They said all this bio stuff that you were talking about, that didn't make any sense to them, because chemical engineers didn't do that in 1975 or 6. So I ended up getting an appointment in what was called the Nutrition and Food Science Department at MIT. But the problem was, the year after I joined, the head of that department, Nevin Scrimshaw, left. So they had 2 associate department heads, and both of them separately decided to give me advice. And their advice is I should start looking for another job. And actually, it wasn't just them. I'll tell you a story. One time, a couple of years ago, Michael Marletta, he was a friend of mine, and he's also now in the National Academy of Sciences. He's chair professor at Berkeley. They were giving kind of this roast about me, and he was going back and telling some stories about what things were like in the late 70s. And I'll just share this one with you from Mike, and it was published. And what he said, just pulling this up, was that one evening he went to a faculty dinner at a Chinese restaurant with me and some senior professors. One of the scientists sat quizzing us while smoking a cigar. And he said, when the older scientist heard my concepts of what I just went over, he blew a cloud of smoke in my face and said, "You better start looking for another job." And then Mike says, "I thought I was in a Fellini movie." So anyhow, I only lay that out just especially for the young people in the audience to realize that, certainly my start was anything but good. But what, of course, happens in science is that people did start to reproduce what we did. You begin to use it, and it became sort of a standard way of studying angiogenesis inhibitors. In fact, it led to proving an isolation of all the key angiogenesis inhibitors that are used today, like inhibitors of VEGF, EGF, FGF, and so forth. But as I'll continue to point out, that was for a while about the only thing it got used for, that and other informational molecules. one of the things that we wanted to do was understand the mechanism better. So Rajan Bawa, one of my students, cut thin sections through the polymer, and this is a non-degradable one. We've also did a lot with degradable ones, but this will get across the idea. So if you do a thin section like this, you see that it's a sheet, and the molecule can't get through. But if it's large. But if you put a drug in, here's a reddish one, myoglobin, just as a marker, what we see is here's the polymer, and here's the drug, and you get what we call a phase separation. This is before any release takes place. But if you release this for a year, and then cut a thin section, what you find is left behind where the drug is are pores. And these pores are large enough so that molecules even millions of molecular weight can get through. But the pores have tight constrictions between them. And they're also incredibly winding and tortuous. So it takes a really long time for the molecules to get through. One way I sometimes explain this to people is it's like driving a car through Boston. And Boston and these systems have what we call, in chemical engineering, high tortuosity. And over the years, like our students would develop all these rules for determining tortuosities and porosities and predicting them. So now we would be able to design systems to release anywhere from days to years to any time in between. And Larry Brown, another one of our students, showed, again, we got an in vivo proof of principle just taking insulin, that we could deliver it from a single pellet for 100 days. So one day, and this is certainly going to be key to a lot of the things that we talk about. One day, Dr. Folkman said to me, Bob, we should file a patent. You know, that might here or any place sound like in 2016, something that people might do. In 1976, when we first started doing this, Children's Hospital, which is one of the best hospitals in the world, they had 0 patents. They'd never done it. So we filed the first one, and 5 years in a row, the patent examiner turned it down. He didn't understand it, but it didn't really matter, because he's calling the shots, not us. So the lawyer from the hospital said to me, Bob, he said, you are costing the hospital an enormous amount of money. He said, just quit. He said, this is a real waste of money. But I don't like to quit. So I started to think, how can we convince the patent examiner, who didn't understand the science, to allow this patent, you know, legally, of course. And as I mentioned to you, when we started this work, everybody said it was impossible, it couldn't happen. And I wondered if anybody ever wrote that down. So something you can do is what's called a science citation search. You can look at your Nature paper, say in '76, and see who cited it and what they wrote. So I did that, and actually I had no idea that this happened, but I did this search in 1982 or 81, and I found this paper. Actually, I found others too. They were all kind of helpful, or a number of them were. But I'll just read it, and this is actually from 5 of the leading polymer chemists in the world at that time. And what they wrote, describing the field, is that generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising. So that was actually, it turns out, a great word for the patent examiner. Results that clearly demonstrate the opposite. So I showed this to the lawyer. He got very excited now, flew down to Washington to see the examiner, and the examiner said, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from these 5 people that they really wrote this. And that's actually true. So we got them. They were all nice enough to write me back that they really, I wrote them. They were all nice enough to write me back that they really wrote it. And we got this very broad patent for really almost any polymer and any molecule. And with that, you know, now it was about 10 years later. So, and like I say, people were using it in science and papers and things like that. But I was kind of sad. I wanted people to use it medically, and nobody was. And then all of a sudden what happened was, around 1983 and 1984, I started getting calls from companies like International Minerals and Chemicals. It doesn't exist anymore, but they were a multi-billion-dollar company in animal health, and they wanted to use it for like animal growth hormones. And the next year, Eli Lilly, which talked about using it for insulin. And I was really excited about this for a lot of reasons. I mean, first they asked me to be a consultant, and so I was able to get, I had a Ford Pinto car for many years, and I was able to get a decent car. And secondly, they gave our lab actually $200,000 a year for research money, each of them, which for a young professor was good. And third, and actually most important to me, was they said they were going to work on it and they were going to make products out of it. And I thought, that's wonderful. That's really what I wanted to see. Well, I got the first two things. They can't really take that away. But the third one, what I saw is they do a couple of experiments a year. And when the experiments didn't work as well as they could, and they never do, they gave up. So that was very sad to me. So about a year or two later, Alex Liebenoff, who was one of my friends at MIT, said, Bob, we should start our own company. So we were able to get those patents back. And 4 of the students who had worked on this in our lab actually went to this little company we called Institutet. And it's very interesting to me, I kind of call this a champions effect. You do this work, you hand it off to, say, Eli Lilly, which is certainly a very good company, but they don't feel the same commitment to it that somebody who's worked on these things for 5 or 6 years of their life or more do. And when stuff started to go wrong, or wrong is maybe the wrong word, but if things didn't work perfectly, well, these guys were working night and day to solve whatever problems they could, technically, and they would. So the difference was, because we had this little company, where people would almost live or die on the success of this, rather than this big company, we were able actually to make a lot more progress. Actually, it's an interesting kind of story. So the science at Institutet was, got us to make some good technical success, but the CEO of the company, wasn't that great. So we actually did a merger. There was a company we were on the 5th floor, there was a company on the 4th floor called Alchemies. And they actually had a great CEO, Richard Kops. And we did this merger where he became CEO of the combined company, and they stopped working on the blood-brain barrier stuff that they'd started on and just on the microspheres. So what's happened is, today, Alchemies has 25 products that are FDA approved or in clinical trials. It's led to all kinds of new treatments for schizophrenia, alcoholism, narcotic addiction, diabetes. They have about 1,500 employees. Some of the products have had, really life-changing effects. You can imagine, again, if you have peptides, people unfortunately don't, there's almost no way to get it to work without microspheres, because it's like if you swallow it or put it in a patch, it's just not going to go into the body, and if you inject it, it's destroyed in seconds. So, but that's not the only time it has an impact. There's a huge issue of what's called patient compliance. I'm going to come back to this again later, which is that people don't take drugs even if they're supposed to. And in fact, one of the biggest areas that people don't take drugs are people with mental health diseases. So it turns out that schizophrenia, just as an example, they've had very, very successful products. Schizophrenia people, I don't know if people saw the movie A Beautiful Mind with Russell Crowe, but he had schizophrenia. He was seeing people that weren't there, if you saw the movie. But the consequence of that is, for a while, the only way they would treat people like that, and that's what they did in the movie, is electric shock treatment. Today, they'd give him Risperdal Consta or something like that, which is a once every several week injection that's based on Johnson & Johnson's drug and our technology. But this keeps going on and on. I mean, there are many examples, and now not just by Alchemies. In fact, here's a picture of many different FDA-approved products based on this that affect tens of millions of patients every year, and they involve schizophrenia like this, but also luteinizing hormone, releasing hormone analogs that are very now widely used for treating prostate cancer and endometriosis. In fact, this initially would last for a month. Now they actually can give them once every 6 months, but there's ways to stop bleeding, prevent pain, also all kinds of things. So this actually was how I started to get involved in this area and realized that, and it made us all feel good that we had done something, and it didn't just lead to, scientific papers, but it was really affecting people's lives. So I'll give you some other stories too. This is the oldest published story, but what we've kept doing in the lab, and I should say, people have asked me this question. When we start in the lab, most of the time, we just try to dream up stuff or, make discoveries that we feel will be fundamentally important. I mean, a company is not necessarily on the radar screen. at all, but sometimes students want that as a career. They want to take their discoveries and they want to create them and bring them out to the world. So we've done this in other cases as well. And the next example that I want to give you is, and I'll do these a little faster, was a microchip. I had this idea in 1994 or 1993. I was watching this TV show on PBS about how they were making chips in the computer industry. And I thought to myself, wouldn't this be a great way to make a drug delivery system? Now, maybe if you spent 30 years of your life at that time working on drug delivery systems, you'd think that about any TV show. But when I saw it, I had this idea of making a chip, and I talked to Michael Sema, who was one of my colleagues at MIT, and the idea I had was that maybe we could make little wells, cover them with a metal like gold, and you could literally put on these chips, in these wells, almost anything you want. And then you could activate them, and when the covers come off, the drugs would come off, and you could activate them one at a time. So we had a graduate student, John Santini, make them. In fact, he started as an undergraduate working on this for a summer job when he was at the University of Michigan. Then he came back and did his PhD thesis on it. And this is the chip. I'm sorry, this is the United States dime. Sorry about that. This is the chip, a top view and a bottom view. This is kind of an old chip, but it gets across the idea. You see 34 wells in the top and bottom. And I'll just show you how they work. Here's one well. It's covered with gold or a platinum alloy. It'll actually stay like that in the body for years. It's hermetically sealed. But if you come along and apply 1 volt by remote control, it's kind of like almost the way you open up a garage door. You can blow the cap off. And when you do, this was from another Nature paper, what John showed is that you could put different amounts of drug in different wells and you get delivery over time at pulses. And here's more like this, almost a pharmacy and a chip idea, that you could deliver multiple drugs at whatever time you want. Over the years, and we did start a little company on this too, and I'll get to that in a second, but one of the things that the company did, and John actually became the founding chief scientific officer, but it actually, because we needed the money to do these things, is we took it from animal studies. There was a paper in Nature Biotech in 2006, to actually human studies. And this may even sound like space-age medicine, but let me tell you a little bit about what was done. Is that what we did is we put the chips in the human body, and you can communicate with them over a special radio frequency called the Medical Implant Communication Service Band. It's approved by both the FCC, that's the Federal Communications Commission, and the FDA. And actually, if you change your mind, let's say, for some reason about what you want, the patient or doctor can put a special computer code in to administer or change the dose. In fact, we've had different reviews, and one time there was a review and somebody said, well, can't somebody hack into the system? So one of the women who actually works at the company, she said, well, I don't think that will be likely. She said, the last project I worked on was the air-to-ground missile. And she said, the encryption here is a lot better. So at any rate, but we do have that built in. And we also built in a bidirectional communications link between the chip and the receiver. The receiver, by the way, could be like an iPhone or something like that. And it tells you everything. Did you take the drug, battery life, and so forth. The first trial was done in Denmark. This was published in Science Translational Medicine. There was 8 patients. And we wanted to pick an area, an idea or a drug which we thought would give a good test. One of the things, whenever we come up with ideas, and I'm sure this is true for many of you, and you write review grants and stuff, people are very good at telling you why your idea won't work. So here, the biggest reason people said it won't work is you'll get fibrous encapsulation. That will block whatever drug you put in. So we said, let's put a pretty large drug in. We picked parathyroid hormone, which is 39 amino acids. We also picked it for another reason now. We felt it could make an important clinical impact. Basically, what happens is, and this is just real statistics, is that if you have osteoporosis, that's the disease that you're trying to treat with parathyroid hormone, that often it's elder women that have it. They're supposed to take an injection once a day. In fact, you don't want constant release. You want pulsatile release, because if you take an injection continuously, if the drug comes out continuously, you get bone resorption. So that's bad. So you want an injection once a day. But what happens is 77% of the women who are supposed to do it, they don't do it. I mean, that's just a fact, even in America. So what was done in the first trial was a small office procedure to do the implant. And what we found was the same pharmacokinetics with actually less variability. I don't think that's important here, but maybe it will be for some other diseases. And these are 3 measures of how well you're treating the disease: calcium and different collagen fragments as daily injections. And just to show you some of the data, this is human data at day 60, 68, 76, and 84. So it's very, very precise on top of each other. This is an early version of the chip. You do get fibrous encapsulation, but it's nowhere near, like it's maybe 1/20th of a pacemaker. If you do histology, there's no inflammatory cells. If you look at these early chips, what you see is there's a chip, there's a computer program, power source, microelectronics, and even a antenna that you imprint right into this. That's how you communicate with it. So again, one of the key themes that was key in terms of trying to translate this into a product was having a patent. And again, the thing that I found, again, when we try to do things in our lab, and our lab is fairly well funded, but still, if we have a grant from the government that, say, is $500,000, that doesn't get you anywhere in terms of trying to do manufacturing and clinical trials. So anyhow, we filed patents. Here we got an extremely broad patent, and I'll just go over that. It's a device for the release of molecules comprising a substrate, at least 2 reservoirs. I won't go through the whole thing, but I want to make a key point. So when you have patents, if you want to have a broad patent, maybe what you'd even call a blocking patent, there's a term in patent language called limitations. And the good thing about this for us is that there aren't hardly any. Meaning, specifically, when you look at the language of this claim, it's for the release of molecules. So that means any molecule comprising a substrate. That means you could use any substrate. And that means nobody else can. And that might sound mean, but see, investors, that's a big important thing. They won't put a lot of money in unless you have really strong patents. At least 2 reservoirs. So that means 2 to infinity reservoirs. Each reservoir has a cap. So you can see the cap has no limitation. It could be made out of anything, and so forth. So this was a very, very broad patent. And because of that, we had, like I say, got the paper in Nature, initial venture investment because of the patent. John, our student, started as a chief scientist. Today, there's investments of over $100 million, and 4 big companies are working with the company, and they're in clinical trials. Now, again, one of the things that I've seen in these companies, and I'll mention this a few times, is that there's ups and downs. And this company, we actually got off to a pretty good start and made some pretty good progress, but everything ended up being much more expensive to do in terms of the research than we ever thought. And some of the investors got tired. They just didn't want to keep money in. So by 2011, even though at one point we had like 40 or 50 people, we had to shrink it down to 2. And that looked like we might be going out of business. And that sometimes happens. Sometimes you take a shot. And our business people, and that's often been the key on some of these, that didn't match the science. At any rate, I had a visitor in 2012 to my office, and he and his wife, he was a very successful computer entrepreneur, software entrepreneur, and he and his wife have this foundation where they want to help the third world. Here's a picture of him and me. That I'm on the left, and you might recognize the gentleman on the right, which is Bill Gates. And one of the things that he and Melinda were very interested in is, could they help women in the third world, right, with birth control? Right now, if women in the third world, if you look at what happens, a lot of times women have abortions. The abortions there are terrible. Sometimes women die, and the birth control systems that exist today are really not that good. None of them last more than 5 years. And the woman has no control over it. Once it's put in the body, it's there for all that time. And what they asked us is, could we, and this is actually right from their website, Gates Foundation, develop a personal fertility control system that might last for, say, 17 years, but that the woman could turn it on and off whenever she wanted. You might ask, how could you do that? Well, here's the chip we're designing. They gave the company so far $13 million. And that totally changed everything. Then Teva gave us a big grant, and now we're back to about 40 or 50 people again. But you might just ask, how would you design this system? And the way it's being designed is, let's assume that we have a chip. It's actually a pretty small chip, and everybody's improved the microelectronics and things like that. But the chip has 200 wells. Each well, say, has a month's supply. So the way you have it set up is the computer program says, the first well, the cap comes off, it goes for 30 days. Then after 30 days, another cap comes off. That goes for another 30 days. Let's say, so if you take 200 of them, that'll go for about 17 years. Let's say, though, the woman changes her mind after 2 months. She just presses a remote control switch, and off it goes. Let's say a year later, she changes her mind again. She can press another button, and it goes back on. That's kind of the design we're thinking about. Also, the other type of thing we're thinking about is really making systems that are smart, like putting sensors on these chips, and maybe someday sensing signals in the human body and telling them how much to deliver. So let me give you the next idea that I want to go over, and that was aerosols. I had a very, very smart postdoc come to see me named David Edwards in 1993. And David, he was like I'm a chemical engineer, but I do like experiments. David was a chemical engineer, and he did kind of mathematical models. And when he came to our lab, he'd never done an experiment. But one of the things that I mentioned to him was, because he modeled the lung, was maybe we could deliver drugs to the lung better. And delivering drugs to the lung is an incredibly inefficient procedure. You're lucky if you get 2% of the drug from the device to the lung. And let me go, and that's bad, obviously, because from a cost standpoint, you lose 98%. But maybe even worse, from a practical standpoint, you'd like to carry an aerosolizer in your pocket. If you lose 98%, that drug's got to be really potent if you're going to have a small aerosolizer. You might ask, why has it been so inefficient? Well, what happens is if you have an aerosol, people always thought that the aerosols had to be very tiny, like 2 microns or so. Otherwise, they'll settle in the back of your throat. The problem is when you make an aerosol really tiny and it's in your aerosolizer, it's kind of almost like wet sand. The surface area is enormous, so it sticks together in the aerosolizer, in the air, and even in your mouth. So by the time you get to the lung, you don't have much. So how did people try to solve it? Well, for years, they tried mechanical engineering approaches, coming up with better designs of the aerosolizers to break apart those sand-like aggregates. And sometimes people made improvements. They could even go from 2% to 3%, which isn't that bad in the sense that you're saving 50% of the cost and you're making your aerosolizer 50% smaller, but still not great. So one of the things that David and I started talking about was everybody up till us started working on the aerosolizer. Nobody ever thought about changing the aerosol. And you might think, how could you do that? Aren't aerosols like water, meter dose inhalers and nebulizers? But a few people by then had made what were called dry powders. And so what we started talking about was, everybody before us, if it was water or a dry powder, used a density of about 1 gram per cc, right, the density of water. And they always made these particles really small. We said, what if we did this? What if we made the aerosols really big, like 10 or 20 microns, but made them very porous, like have a density rather than 1 gram per cc, 0.1 gram per cc. And let me just give you, and our thought was that would make them easier to aerosolize. Also, they might be less prone to phagocytosis. And so the idea is that you'd get much more in because they wouldn't stick together. Like wet basketballs, for example, aren't going to stick together like wet sand. Also, they might last longer because you can imagine if you inhaled the big aerosol, macrophages are going to normally clear them, but now it's like eating a big meal rather than a small meal. Even though it turns out the mass is the same, but the size it sees is not. So at any rate, we had several students, Justin Haynes, Giovanni Caponetti, Jeff Herkash, and then Noah Lotan on sabbatical from the Technion. And we made some aerosols. Here's a scanning electron micrograph of one. I'll give you a sports analogy. This may not be apt in Sweden, but before us, if you looked at the sports analogy, if you looked under a microscope at regular aerosols, they'd look like little golf balls or little baseballs. I don't know if people have wiffle balls here, but they're very porous. And that's what these look like. So they totally change their nature. But that changes everything. So all we did is change the geometry. But what that meant is you get 10 times more in an in vitro experiment, mimicking the lung. And if you did an in vivo experiment, these are different lobes of the lung, again, you get 10 times more in. If you do a human experiment, we actually made it last for 3 days. So again, we filed for a patent. I won't do this every time, but this is one of the themes that I would say is important if you want to get significant funds to develop these for products. And our densities, like I say before, people used the density of 1. Ours were generally 0.1, but we've made a cutoff of 0.4. And the particles would be larger. And again, it's any agent, so it was a very broad patent. And what we did is the initial studies were done in the labs. We published a paper in Science this time in 1997 showing it. We started the company in 1998, and everybody wanted to use this. So there were deals which David and Terry McGuire negotiated with Eli Lilly, Glaxo, and Pfizer. And because we hadn't changed the chemistry of the drug at all we did is change its geometry. That enabled us to start 2 human clinical trials within very few months of starting. And David decided he didn't want to do this forever. So about a year later, he said, well, maybe we can merge it with somebody or sell it. And actually 4 companies wanted to acquire it, and the investors made over 1,000% IRR. That's an annualized return. So I think it's the highest in history, or one of them. But, and we'll develop it further. But interestingly, that led to several other things. David wanted to help people in the third world, and myself as well. And so, again, when we first started suggesting this idea of large aerosols and wrote grants to the NIH, we got totally rejected. They said, this is impossible, it won't work. But now, showing that it was working in human clinical trials and products, everybody knew it does work. So you could actually now get a proof of principle, and he began using this to spin technologies off for tuberculosis and so forth, with funding from the Gates Foundation. And then a new company started, Civitas, to make this for Parkinson's disease. And they got acquired recently for quite a bit of money as well. I thought I'd give you 3 more quick examples. They're all sort of different. Again, all done by our students. And proteins, of course, people are monodispersed. Like people know, insulin is always the same molecular weight. Nucleic acids also monodispersed. And all of these have been sequences. In fact, Nobel Prizes have been given for sequencing this and this. Polysaccharides, though, actually are more complicated. They're actually polydispersed. And let me just give you some examples. If anybody's looked at heparin, which is a blood thinner, is polydispersed, right? There's heparins that are at that 10,000 molecular weight, 11,000, 12,000, and so forth. And glycoproteins. The protein part is the same, but the glyco part is not. So one of the things that we did over time is think about maybe we could make a way to rather make what I'll call a glycomics company. And the way we did that, our thinking is in these polydispersed units, we could fractionate them to find what we call specific glycoforms, right? In any, I'll make this up, but let's say if you had heparin, there's 100 different molecules. There's probably more. It's a mixture, even though the FDA approves it. But you could fractionate out each one of them. And then what we did is we actually did develop the first sequencing approach to complex polysaccharides. This was actually done by 2 of my students, Ram and Skaskaran, who's now a professor at MIT, and Ganesh Venkatraman, who's now running the company that I'm going to talk about. And what we did is we used the molecular scissors. These are what we call heparinases that we'd actually isolated and published in Science for actually different purposes. A combination of molecular scissors, so that you cut them in different places, you use mass spec to identify it, and then sort of mathematical modeling to piece it together. So for the first time, and we got patents on this, we could actually sequence complex polysaccharides. And what that led to, again, when we wanted to make products, are new heparins, like low molecular weight heparins, other complex molecules, and new glycoproteins. So again, Ram and Ganesh wanted to start a company. This is also, to me as a professor, something very important. And a lot of our students, they have this dream, like I said, of seeing their work really get out to help people, and they have this dream also, maybe it's part of the MIT culture, of often starting companies. So we started with them. It went public in 2004. There were major investments so far by Novartis, Baxter, and Mylan, all deals. But what was also interesting is the first low molecular weight biogeneric ever approved by the FDA was actually a momentous product, even though many larger companies like Teva started about 3 or 4 years before, but they didn't have this sequencing technology. So we did. So we got that approved, and it was actually based on purely analytic data, not clinical trials. It was actually the largest syringe launch in history. And then Momenta did the same thing again last year with Copaxone. That's a multiple sclerosis drug. Though I also wanted to say, and here's just a picture of the product, but there's a whole other story that I want to tell you about this. Sometimes people would say, well, companies, it's not, they don't do good things and stuff like that. Well, in 2008, actually, I was chair of the FDA Science Board, their highest advisory board, and I got a call, and so did Ron, because they knew him too, from the FDA, because what happened in the United States, and it may have happened here too, is in 2008, a lot of people started dying from heparin. There was contaminated heparin coming over from China, and these are just some of the headlines all around the United States. And actually, it went up to have tainted heparin, not just in 11 countries, but 22 countries where they found it. And it was like a major disaster, people dying from this. So the FDA called us up and they said, could we help? So what we did is, using the sequencing technique, we took that heparin and we published 3 papers. Actually, it was kind of interesting, because I think this is the first time in history, at least to my knowledge, where you see paper from MIT, the company, Momenta, the FDA, and the CDC, that's the Center for Disease Control. And what these papers showed, by myself, Ram, and others in the company, is first, here's the contaminant, oversulfated chondroitin sulfate. That was the contaminant causing the adverse effects. And 2 papers in the New England Journal of Medicine tying this contaminant to this outbreak of adverse events. And after this, there were no more deaths. So this is actually using the sequencing technology to solve a major world health problem. So I'll give you 2 more quick examples and then make some summary comments. This one is kind of recent. I had another student, a graduate student, Armin Shari. He was an undergraduate at Stanford. And one of the things we started playing around with was, could we come up with other ways of delivering drugs to a cell? And as you know, right now there's a lot of techniques for delivering drugs to cells. And this is actually important, for example, if you're trying to engineer cells that, say, kill cancer cells or other things like that. So how do people do it? Well, sometimes people do electroporation. Sometimes they do liposomes. Sometimes they do nanoparticles. Sometimes they do cell-penetrating peptides. But each of these has some drawbacks. You can, and I'll show you this more, you can damage the payload or get toxicity with some of these. You can get chemical modification with some of these, off-target effects with some of these, and it also often is hard to get out of the endosome. So Armin, my collaborator in this study and his co-advisor is Klaus Jensen. Klaus actually is an expert in microfluidics. And Armin was playing around, again, this is almost a serendipitous discovery, but he was playing around with microfluidics by shooting cells through channels, constricted channels. And I'll just show you a video. So this is one of the things he made. So we put the cell in, and notice how it goes through this constriction. It'll just keep doing it. And we can actually do this now at 1 million cells per second. This was published in PNAS. And just to show you a little bit more, in that paper, we tried to re-engineer human fibroblasts, basically to see if we could make iPS cells. If you use a cell-penetrating peptide, you get this type of colony count, nucleofection, that's electroporation, probably one of the better forms of it. You get better. And with the device that we made, and now we have devices that can have many channels that have 1 million cells per second, you get this. In fact, in 2014, Scientific American came out with their top 10 advances of world-changing ideas. CRISPR finished first. This finished second. I felt they kind of had the order wrong, but we said that's still okay. And Armin wanted to, he started doing a postdoc, and then he said to see me someday, one day, I loved talking to students. I said, well, what's your dream? He said, my dream is I want to make this company great, and I want to create a company out of this. So we did, and he started this company. One of the things that they did is work out ways of delivering, say, siRNA or other things to immune cells. But here you see some improvements compared to with microfluidics compared to nuclear infection. Again, here you see a significant improvement. But what was really striking is the damage that electroporation causes. This is a transcriptome, which gives you messenger RNA. So if you look at the cell before you do anything, this is what you get. if you do electroporation, and this is actually what gets used clinically a lot of times, you can see the changes. But if you do the squeeze technology, it's not identical to the control, but certainly much, much closer to the electroporation. And he started this company, and it actually got Huffman LaRoche was very excited about it and gave $500 million deal, not all up front, but a significant amount up front, with the idea that you could use the squeeze technology to engineer B-cell presentation, and the hope was that this could lead to better ways of helping, say, CAR T cells. And it's sort of a limited area, but very exciting. And the very last example I want to give before I summarize is a very different thing. This also came from the Gates Foundation. So one of the things that Bill and Melinda are very interested in realize is that, and I mentioned this before, is patient compliance is terrible. It's bad in our world, 50%, say in Europe or the United States. In developing countries, it's even lower. And one of the things that they've said, and this is from the World Health Organization, is increasing the effectiveness of adherence interventions may actually have a far greater impact on the health of the population than any improvement in specific medical treatments. So in other words, if you could somehow get people to take their drugs that already exist, that might be more important than having even new strategies to make drugs. But how can you do it? And that's been a tough problem. So the difficulty is, and I'll just give you an example or two. They said, take malaria. They've done calculations. Some of the people there, like Phil Eckhoff, if you could keep the level of ivermectin, what won the Nobel Prize last year, over 6 nanograms per mil for 2 weeks, their calculations are that that would get rid of malaria. But people won't take the drug more than the first time very often. And the problem is, as you know, even though you can give injections, people don't want to do injections. But could you ever design, and this is the question we ask, a pill that could last longer than a day? So normally, you're out of the gut in a day. But one of the thoughts that we had is if you take a standard capsule, it does this, right? This is the stomach, the intestine. There are ways that we and others have worked to slow it down, like bioadhesion, and that cartoon is shown here. But usually at most, that gives you an hour or two more. Doesn't really change things fundamentally. But the idea that I had with Gio Traverso, Gio actually is one of, I think, an interesting person. He actually got his PhD with Bert Vogelstein, who's a top molecular biologist at Johns Hopkins, an MD in gastroenterology, and then he came to our lab, which does more chemistry and engineering. And one of the things he and I started talking about is maybe we could do something radically different. And that is the following. What if we could make a pill that you could swallow, and let's say we put it in a capsule, but that capsule will, when it gets to the acidic stomach of the pH, will dissolve. So you sort of compress this system up into the capsule. You don't even realize what's in the capsule. But let's say it's something that you can put in the capsule. Now you get into the stomach, and it opens up into a shape. And the idea of the shape is that the shape is so big that it can't go through the pylorus. That's the hole between the stomach and the intestine. But it's so open that food can pass. And also, we can tune its degradability from other things we've done. So this is the idea. Let me just show you a picture of what we can do. So this is sort of the idea, and it would just stay there. Now that might seem like science fiction, right? So here are the challenges that we would face. Can we control drug release rates? Can we deliver the drug for a long period of time? Can we control polymer degradation? All of these we'd solved before. All those are based on other things we've gone over. But the last two, could we control polymer shape? Can we do it safely and relatively inexpensively? Well, we actually did publish some papers on controlling polymer shape. We created sort of the first degradable shape memory polymers. We published one of these papers in Science, where we used temperature as a trigger, and another in Nature, where we actually used light as a trigger. I'll just show you just a proof of principle of it. So one of the thoughts we actually had was in minimally invasive surgery. Maybe we could make something like a string at room temperature, but when it goes into body temperature water, it changes to a coil, like a stent. Could be anything. So here's a picture. You see this. So again, this was a string at room temperature, but as soon as it went into body temperature water, let me just do that again. It's a string at room temperature air, it's body temperature water, and we programmed it to change into a coil. So we could easily change it to another shape. Here's a second example where actually, and this is the intent might be in minimally invasive surgery. Let's say you want to tie a surgical knot. So if you're outside the body, it's pretty easy to tie a knot. Like I could do it. You might not want me to do it, but I could do it. But like inside the body, if you want to tie a surgical knot, it's really hard, like right in the stomach. So let's say we might make the knots tie themselves. What if we, just to prove a principle, so we have this loose knot like a lasso here in body temperature room temperature air, where we put it into body temperature water, it just tightened. Again, let me just do that. Here's the loose knot, air, now water, 12 degrees higher, tightens. So we had that ability to do this. So could we, and we even made, you could even do this by making a superelastic polymer, and we published an article on that in Nature Materials last year. Finally, can we do it safely and relatively inexpensively? Well, here are a couple of the shapes that we came up with. This is the one I mentioned before. We also had this star shape. And we could either tune the degradability of the whole thing, or we could have specific linkages that we can make break. But basically, we could do that. And what we actually did is, with Geo and many people, is we actually solved that problem that I wanted to mention that the Gates Foundation asked us. We haven't published this yet, but we will soon, actually. But here you see, normally if you give ivermectin in pigs, it goes down to 0 pretty quickly. But here with this system, we gave it to 3 pigs, and we're able to keep the level well above 6 nanograms per mil for 16 days with just a single capsule. That's not the only one we did. There are also, that's for malaria. This would be for TB. And again, we delivered a capsule here for 10 days. And again, I see no reason why we can't go almost any length of time. They've also done studies, Geo and the team at MIT, where they've looked at safety in over 200 pigs. And again, these are all clinicians looking at it, like Geo and others, and Andrew Bellinger. But basically, they saw no adverse events. Pigs had no clinically apparent symptoms, no obstruction, no perforation, no change in appetite, no weight gain, no change in weight gain, no change in stool frequency. And actually, the clinicians found no mucosal injury or gastric ulceration, and they did all types of endoscopic evaluation and autopsy. So again, several of the students wanted to start a company on this. And these are all our students, so that after they did their theses or their postdocs, they went to the company, and but they got Amy Schulman, who's got a great business background, to be a CEO. So that's just starting, but they've already raised some significant funding and will raise more. So those are the 6 stories. Over the years, actually, I think our students, with my help, we've probably done about 33 companies, starting with Enzotech and Alchemies in 1987. There's probably close to 1 a year. This was microchips, but they keep going. Actually, this was the next thing, but one of the ones I wanted to highlight, and in fact, the ones I've talked to you about are not necessarily the most successful ones. One of the most successful ones is Moderna, which actually was founded with Ken Chen, who's actually at the Karolinska, and I think they've given a grant to the Karolinska, or have a grant with the Karolinska. And that, here, the CEO's been just unbelievable. He's raised over $1 billion. This is a whole new kind of RNA and could lead to new therapies. And again, one of the big challenges is delivery to the right cells. And they've just started clinical trials. And then you see others like Squeeze, Lindra, which I just mentioned. The latest one we haven't even announced yet, Siglion, that's based on some papers we published in Nature Medicine, Nature Biotech, and Nature Materials just in the last few months, which is a new way to encapsulate islet cells for treating diabetes and other diseases. I thought I'd conclude just with some lessons that I've learned. On the science side, I think what's worked well for us is having what I call a platform technology. Lots of things you're going to do aren't going to work, but if you have a platform technology, and that actually helps you in a couple ways. One is, if some things don't work, others might. 2 is that rather than, like, say, just license it to a big company, let's say we license everything to Merck. Merck's a nice company, but if you'd licensed everything to Merck, they would just use it on Merck's drug. If you have a small company, that company could work with Merck on their drugs, Pfizer on their drugs, Astra on its drugs, and so forth. Generally, we've made products almost every time these have come out of papers. We've never held up the papers. I always consider the papers to be key. So they've been published generally in top journals. But we have gotten patents every time. And those patents, if we didn't have them, I don't think the investors or the companies would make significant deals, which unfortunately it takes to manufacture and get things to the clinic. Also, we've shown in vivo proof of principle. Medicine, in my experience, is a lot more expensive than starting a company, say, in the computer area, IT. So you really want to have an in vivo proof of principle to lower the risk. And also, to me, one of the wonderful things being a professor is I want to see my students live their dreams, and many of them have wanted to do this. And every one of these companies, they've had leadership roles in. A couple other things, though, that I thought I'd mention. Since medicine is so expensive, figuring out a whole lot of what I'll call non-scientific things also ends up being really key, maybe sometimes more key. So having a good regulatory strategy is really important, FDA strategy. Having a great management team. In fact, I hate saying this as a scientist or engineer, but I would like to think that every time we did a company, the science was the most important thing. And it is important, but sadly to me, is the business people play such a critical role. And when I correlate the success, if you wanted to look at it financially or even sometimes clinically, it's often been because of the CEO and the management team and their ability to raise funds. Because unfortunately, the medical area is so expensive and failure is so common. Something like 6% drugs get approved. Having a CEO who can raise a lot of money and weather these storms, that's just been so critical. And so that's been key. And tied to that is having patient and well-funded investors, which usually does mean venture capitalists. But I also put a few other things. If you have novel therapeutics, and we're seeing a lot of novel therapeutics now, like siRNA, mRNA, CRISPR, delivery, which is something we talk about a lot, that's an incredibly difficult problem. That's probably been the biggest problem for almost any of these therapies. So if you want to have a new technology, you want to get a proof of principle. So it's good to pick situations where delivery isn't that hard. Alnylam, which is, for example, an siRNA company that I've been on the scientific advisory for from the beginning, what they've done, which has enabled them to be successful, is to sort of deliver everything to the liver. That's sort of an easy thing. So 100% of their technology is aimed at that, and that really has lowered the bar, and they've done well. I think we'll see some products approved by them soon. Also creating a great culture where the scientists are excited and using advisory boards well. At any rate, that's kind of what I wanted to go over to you today. I realize it's been a little bit different lecture. I'm more than happy to answer any questions. My feeling is that these systems, we're really trying to take it from the academic place to the clinical place, and that these companies have just played an absolutely key role in enabling that to happen. So anyhow, let me stop here and answer any questions, and thank you so much for having me.